uh, ingestive nutrition and others. Next, third one, the fungi in which nutrition to observe. In the fungi nutrition to observe. And next, coming to the or fungi plants. Here, fungi plants if bacteria or not. That is the one controversy is there. The fungi plants are the bacteria or not. There are reasons to say they are not. Fungi comprise a separate major group of organisms which differ from plants in origin, direction of evolution and organization due to adaptation to different mode of primary nutrition. That one is called observative. In adaptation to observative mode nutrition, the higher fungi have evolved. First character, non-motile life embedded in food supply. This is the first adaptation. The second adaptation, mycelial organization that combines maximum surface of contact with the free movement of food and protoplasm through the mycelium system. The distinctness of fungi from plants and animals has been supported by studies of cytochrome C, a component of the terminal respiratory chain of enzymes in aerobic organism. Fungi, as Martin remarked, may reasonably be treated as discrete major taxonomic unit. The slime mode. The slime mode defy the distinctness of the kingdom in both nutrition and organization, offer free choice of treatment, that is the fungi. Position and peculiar animals. Okay, here these fungi acrophilic, heterophilic, eukaryotic, thallophyte. This one two different scientists to explain to the different ways. The branches of botany deals with the fungi is called that, and you can study the fungi, that name is called mycology. And the scientist who concern with the fungi is called a mycologist. With the scientist to mycology, so concern fungi is, that is a mycologist. Mitchell, father of mycology. Who is the father of Mitchell? P.A. Mitchell, known as father of mycology. E.J. Butler, referred to as father of Indian mycology. Mitchell is the world mycologist, father of mycologist Mitchell, P.A. Mitchell. Our Indian mycology name is called Butler. Butler referred to as father of Indian mycology. Fungi are non-green in color. Here color is not particular. Color is, but that's why here chlorophyll is absent. Why? Color is a non-green color with the capacity to live in all kingdoms of environment. Another one, where the domain of fungi. The limits of fungi are not always well defined. There are group which are retained in fungi only by tradition. Example, the Myxomycetes. Myxomycetes meaning slime mold are organisms studied by mycologists. They are also studied by zoologists and placed under mycetosa. The cellular slime mold, that is, which are also organisms and therefore should be classified into protozoa. This is the domain of the fungi. What is the characteristic features of the fungi? See, this is a difference in between the algae and fungi. What characters is present in fungi? Fungi are too diverse to be easily defined. The main characteristic are the following: adopted from Ainsworth scientist in 1973. The based here main important the difference in between the identification of algae and fungi. How to identify the algae and fungi? Here the algae process chlorophyll, which in some of them is masked by the other pigments such as brown color, red color, blue green color, yellow color, like this. This is the algal pigmentation. But when the fungi is there, in that fungi, lack of chlorophylls are present. Students, did you understood my lesson? Yes or no? Tell me anyone. Did you understood? No? Anyone to respond? Okay. Another one. Second character, with help the chlorophyll, they are able to synthesize their own food and that is called autotrophism in their mode of nutrition. Okay? They prepare the own food material in where in algae. But you can observe the in the fungi, here chlorophyll is lacking. Absent chlorophyll, no? That's where the fungi are unable to synthesize their own organic food. That's where they get it ready-made source, either by living as a parasitism or sporophytism. Why? The fungi can't to prepare food material own. That's why they living by either parasitism or saprophytism. For this reason, fungi are referred to as heterotrophism in their mode of nutrition. This is the most important character for the fungi and algae. Fungi, algae is the, they prepare own food material. Why chlorophyll is present? That's why autotrophism. But here, fungi, they do not to prepare the own food material. Why? Here, lack of chloroplast is there. That's why they did not prepare the food material. This character depending the other living organism, the other living parasitism or saprophytes. For this reason, fungi are referred as heterotrophism in their mode of nutrition. Next character, algae thallus. You know, algae made up of the plant body is called. Shweta, did you understand my, Shweta or anyone to tell me? Did you understand my class? If you observe the PPT? Yes or no? Anyone to respond? Anyone to respond? మీకు అర్థమవుతుందా?
Okay, madam. Yes, madam.
Yeah, okay, sure. Next one, uh, thallus. Coming to the thallus. The algal thallus is composed true parenchyma cells. The unit of structure is called thallus. You know, undifferentiation plant body is present in algae. That name is called thallus. Here, but what type of tissue is present? Mostly the thallus made up of parenchymata tissue. Okay. Coming to the fungi. Here, the fungal thallus, which is termed as mycelium. See, the algae thallus is, algae plant body is called thallus. Fungal plant body name is called mycelium. Is not formed by division and differentiation of the cells, but composed of a false tissue is called. Here, differentiation of the cells composed of the false tissues are present in this mycelium. That's where the false tissue name is called pseudoparenchyma. Here algae is the parenchyma is there. But in fungal, the false tissue made up of the thallus, that's where this is called the pseudoparenchyma. Thus, the unit of structure of a fungus thallus is not a cell, but that name is called hyphae. What is the name? It is not cell, but that cell name is called hyphae. This is the plant structure. Coming to the cell wall. What type of cell wall is there? Here, another important, fungi thallus are in the substratum and amoeboid, plasmodial amoeboid or pseudoplasmodial, or in the substratum and unicellular or filamentous, that is called the mycelium. The last septae are non-septae, typical non-motile, with protoplasm flow through the mycelium. But motile state, only zoospores you can maybe occur only motile zoospores. This is the fungal thallus. The fungal plant body, not for thallus, plant body mycelium like this. Coming to the cell. What type of cell is there? Here, the cell wall in the algae made up of cellulose. Almost the fungi cell wall, sorry, algae cell wall made up of cellulose. Fungal cell wall is made up of hyphae or cell wall composed of cellulose, are often called chitin. A well-defined typical chitinized, that is called cellulose in oomycetes. Other class, the cell wall made up of chitin. Mostly chitin is the dominate to made up of cell wall in fungi. Okay. Next one, what is the nuclear status? After that one, I'll explain to the food. Nuclear status, eukaryotic multinucleate. This fungi is the eukaryotic multinucleate. The mycelium being homo or heterokaryotic. Mycelium being homo or heterokaryotic, haploid, dikaryotic, or diploid. The last being usually of limited duration is present nuclear position. Okay. Coming to the food material. Here, for algae food material chiefly made up of the starch. In algae, you can observe the starch is commonly represented. But in fungi, the reserve food material invariable accumulated in the form of glycogen dots or non-starch material. In fungi food material, reserve food material, what are the reserve food material is there? Glycogen dots or non-starch material. Okay. Next one, the algae grow in habitat where sunlight is available. The algae to grow, it's compulsory sunlight is available. Sunlight is present. Where sunlight is there, the algae to grow. But in fungi, opposite. They generally have grown in dark and dim light. See, fungi meaning just like mold, booju pattadamu antaru chudandi. Yes or no? That one. Okay, when the air is not come, light is very less, dim light and darkness, in that nature, the habit of the fungi, the generally that habit is present. Another one, another character differentiated, the algae grow in water or in a damp soil, sometimes epiphytic or rarely endophytic. Here, they have grown in the wide variety of habitat. What are the habitat as there? Parasitism in the tissue of plants and animals. Always fungi is the parasitism. Yes or no? They can't to live in the alone. They depending other organism. That's why this is the saprophytic or parasitism. They, the ready-made source either by living as parasitism or saprophytism. That this is not to own the, they grown in the general grown in the wide variety habitat, that is a parasitism or tissue plants and animals. Coming to the reproduction. Reproduction here algae apparatus increase in complexity from the simple to the higher forms. In this one, fungal reproduction, there is a progressively and gradually simplification and ultimate illumination of the sexual apparatus from the lower to higher fungi. This is the reproduction of the fungi. Next one, life cycle, simple to complex. In that one also, sexuality, asexual, sexual, homo and heterotalism. Sporocarps also microsporic, macroscopic, scopic and showing to the limited tissue parasitism. Distribution cosmopolitan. We shall examine their character in the some details. Here, similar between the algae and fungi. Fungi and algae both prefer to live moist environment. In fact, one of benefit algae derive from their symbiotic in like relation. In the how to made up of lichens, both algae and fungi combine with to form to settle down. That part is called lichen. In that lichen, algae is the one part from their symbiotic lichens. The relation with the fungi is their ability to survive on land in a moist environment. Both here, algae and fungi are talophytes. And the plant body did not differentiated into the root and shoot system and leaf system.
హలో మేడం. హలో.
వినిపించట్లేదమ్మా? హలో.
హలో మేడం. వినపడట్లేదు మేడం.
వెయిట్ 1 సెకండ్ 1 సెకండ్. డోర్ దగ్గరికి ఇప్పుడు ఇప్పుడు ఓకేనా? హలో.
ఓకే మేడం.
ఓకే. హియర్ బిట్వీన్ సిమిలర్ ఇంతసేపు నెట్వర్క్ ఇష్యూ ఉన్నట్టుందమ్మా. నెక్స్ట్ వన్, similarity between the algae and fungi. What are the similars are there? Fungi, algae, both prefer to live in most environment. In fact, one of the benefits algae derive from their symbiotic relation is the algae and fungi. That relation is called the lichens. And both are to combine with together to form the lichens. Here, fungi is ability to survive on land in a moist environment. In algae, water is must know. That's why algae is not to survive, but algae to depend the fungi to survive the soil moist, but the algae to produce the food material. Both are benefits. That is a symbiotic. Both here, algae and fungi are thallophytes. You know, that is called the undifferentiated in the root system, stem system, leaf system. Both algae and fungi placed together in the division, that is the cryptograms. Cryptograms meaning, what is the cryptograms? Non-flowering plants. This is the thallophytes. Both are thallophytes. In fungi, you can take anyone, algae and fungi. Both are thallophytic. But the difference in the thallus, the algae plant body name is called thallus. Fungal plant body name is called mycelia. But both are the thallophytes of cryptogams. With the expansion of blue-green algae, majority of the algae and fungi are eukaryotic. Okay. Only blue-green algae, expect blue-green algae, all fungi, the cell structure is called eukaryotic structure. Fungi, algae, both general examples possess haploid nuclei. They have only single copy each chromosome. Yes, very important. Why? Haploid nuclei is there. That's why the single copy, each and copy of single, a copy of each chromosome is a single. By contrast, diploid organisms such as humans and most mammals have two copies of each one. Okay, two copies of each one. But here, this one is a main important is the here news applied, that's the single copy. All the diploid conditions are there. Two copies of each one is to present. Another thing is both algae, fungi process cell wall made up of polysaccharides. Chemical nature of cell wall, various algae and fungi. Symbiotic members are present in both group, that is which algae with animals, fungi with to their higher plants. Between the algae and fungi as called lichens. Just now I'll explain no lichens. Both groups both groups can reproduce by the vegetative production, by fragmentation and fusion. Next one, both algae, these both algae and fungi can reproduce asexually by various motile and non-motile plants. Next one, sex organs are naked in both groups. No protective covering sex organs in both algae and fungi. These are the similar character. Before I'll explain to the what is the difference in between, I'll explain. But here both similar. Why both are the to explain? This one is the characteristic is the most important. Coming to the general account, Some of the most important characters, general. First one is the occurrence. Second one is the thallus. Third one is the differentiation of mycelium and cell structure, nutrition, heterothallism, homothallism. Okay, these all characters are important for fungi. First, occurrence, where the fungi is there. See, you can observe the picture. These are the fungal. These are the mycelium, unicellular mycelium. This one. Occurrence, fungal is main important cosmopolitan, occurs in air, water, soil, and plants and animals. Where you can observe the fungal is there. That is the air, where the air is there present, water, soil, plants and animals. They prefer to grow in warm condition, humid places, dark places, and keep food in the refrigerator to prevent bacterial and fungal infections also. This is the occurrence of the fungi. What is the thallus? See, the thallus of fungi. The thallus of fungi usually consists of microscopic tubular filament called hyphae. What is the hyphae? Listen careful. The thallus mycelium is the name. What is the mycelium? First, before mycelium, what is hyphae? You can by hand it. The thallus of fungi usually consists of a microscopic tubular filament is called hyphae. Hypha is the single. Mass of hyphae consisting the thallus is called mycelium. Did you understand? What is the hyphae? What is the mycelium? Hyphae is the fungal usually consists of a microscopic tubular filaments. Microscopic tubular filament is called hyphae. The mass of hyphae consists the thallus name is called mycelium. Did you understand hyphae and mycelium? Anyone? Yes? చెప్పండి, మీరు ఇంటరాక్షన్ అవ్వకపోతే కష్టము. Did you understand hyphae and mycelium? Sorry, next. Another one, the hyphae are always branched, rarely unbranched, very rare unbranched. Always hyphae is a branched, rarely unbranched, and the branches ramify on or inside the substratum to form a three-dimensional network. One cytoplasmic streaming in fungal hyphae is unidirectional towards the tip. Where the growth takes place, hyphae grow entire at their tips. Hyphae always grow entire at the tips. And the key to the fungal growth lies in the apex. There is built-in mechanism at the hyphal apex for verifying the width and shape of the apex. It is the site of active differentiation and nuclear divisions are present. The thin and plastic hyphal tip 50 to 100 millimicron in the apical region is the zone of elongation and is filled with the protoplasm. The portion behind this vacuolated and incapable of elongation, but helps in growth by synthesizing the cytoplasm, which is transported to the tip. Lateral branches arise behind the apex in the acropitial order. Each of these daughter branches behaves like the parent hyphae and forms secondary and forms the secondary and tertiary branches behind their crop tips. And that develop, you can observe the hyphae. See, this is a hyphae, how to develop the tips. The hyphae may be separate or uniseparate. The uniseptae have nuclei created in cytoplasm. Hello? Good morning, ma'am. This condition is known as xenocytic condition. Very important. What is the xenocytic condition? The hyphae may be septae or uniseptae. The uniseptae hyphae have nuclei scattered in the cytoplasm. This is called this condition is known as xenocytic condition. Even in the aseptae hyphae, septae region and to delimit the sex organs, such septae are formed as the result of changes in the local concentration of cytoplasm and are free called adventitious septae. The other type of septae is primary septae and form in occasion with nuclear divisions are there. The between hyphae, the septum is formed by annulus of wall material, which investigation plasma membrane and grows centrifugally inwards. And the And another thing is the plasma membrane is continuous around the septae. These two septae are there. That is called diplos, dip, doliphore septum. Very important. Doliphore septum in the sometime I'll explain. And the how many types of thallus are there? First types is the unicellular thallus. Unicellular thallus, here in some of lower fungi thallus is more or less spherical, single called structure. At the time of reproduction, it become a reproductive unit. Such fungi are called holocarpic. In the unicellular holocarpic holoforms, the mycelium is absent. For example, syncriterium. Some holocarpic fungi producing bud cells in succession, and these remains attached to each and one other. That cell, so that chain of the cell is referred as pseudomycelium. That one is called the pseudomycelium. And next one is filamentous thallus. What is that filamentous? In most true fungi, the thallus structure is called filamentous, composed of a hyphae. Vary in race, in the shape and size. Branching of hyphae is dichotomous on the basis of presence or absence of septae, uh, of hyphae, mycelium fungi. These mycelium fungi are two types. One is the non-septae or aseptae fungi. Second one, septae hyphae. What is the non-septae fungi and aseptae? Here, non-septae aseptae mycelium contains numerous numerous nuclei lying in a common mass of cytoplasm. Common mass of cytoplasm. And without here without cross walls in the hyphae. For example, you can observe to the oomycetes and zygomycetes. This is the non-septate one. Another thing such as a condition is called, that condition name is called xenocytic condition. Very, very important. What conditions are there? That condition is called the xenocytic conditions. And this, however, the septa is, the septae may be lie down, the septae lie down at the time of formation of uh, at the time of formation of reproductive organs to delimit them from the rest of vegetative hyphae. Therefore, called that name is called pseudosepta. Best example for pseudosepta is allomycetes. All allomycetes under in this one. Next one, septa hyphae. What is the hyphae? What is the septae? Hyphae or septae, and hypal segments may contain one, two, or more nuclei. That is example for Ascomycotina, Basidiomycotina, and Deuteromycotina. There are two types of septae are present. First septae name is called primary septae, and second septae name is called adventitious type. What is the primary septae? Primary septae are formed in association with mitotic or meiotic nuclear division. What is the primary? That is the septae, mitotic and meiotic divisions, but meiotic meaning meiosis. And they separate their nuclei, daughter nuclei. This type of septae are found in, you can observe to the Ascomycotina, and next one is the Basidiomycotina. Their asexual stage, you can observe this primary septae. Okay. Next one, secondary septae. What is the secondary septae? Primary septae are formed in association with mitotic and meiotic nuclear division, and they separate the daughter nuclei. These type of septae found in both Asco and Metido. Next, adventitious septae. Adventitious septae are formed in the absence of mitosis or meiosis and occur especially in association with the change in local concentration of cytoplasm. They are found in lower group of fungi. That one is called Mastigomycotina and Zygomycotina. Both are the lower or starting classification. Zygo and Mastigomycotina. Okay, this is the hyphae septae. In the fungal tissue, what type of tissue to made up of these hyphae or mycelium? Here, these are formed by branching, interweaving, anastomies, and coiling of hyphae. During these events, septation occur and the hyphae divide and become short-called and compressed. Fungal tissues are thus relatively simple and limited in variety. They differ mainly in the compactness of organism of the hyphae and degree of division into short cells. These are two types. One is the prosenchyma and second one is the pseudoparenchyma. What is the prosenchyma? In which compact hyphae are loosely woven and can be recognized. That is the Prosen hyphae. Pseudoparenchyma. What is pseudo? It consists of closely packed hyphae which under sections resemble the parenchyma of higher plants. The hyphae divide into short cells which by mutual pressure become isodiametric. Placent, uh, placent, placentation tissues in higher fungi are formed in some the somatic structure such as mycelial strands, rhizomorphism, pseudo-rise, sclerotida, pseudochlorida, stromata. These are general structures are divided into anatomical region and outer and hard rind called cortex made up of pigmentation, that is the pseudoparenchymata, inner soft hyaline prosenchymata core is called medulla, and plasmodium also in the presence in this mycelium. Plasmodium, the plasmodium, which is multinucleate mass of protoplasm without a cell wall, represent the thallus of myxomycetes and plasmodiophoromycetes. Another type of thallus is the amoeboid plasmodium, formed by aggregation of unicellular, uninucleate, naked cells. The aggregation cells look like a plasmodium. This is the pseudoplasmodium. These are found in two groups of organisms. The acracial, libricial, which are placed alongside the myxomycetes, and plasmodiophoromycetes in the subdivision of the myxomycota. These all are the, what is this one? Total, what is the thallus organization like this only. Coming to the, and I'll explain this one. Coming to the mycelium. Placental tissue, just now I'll explain, no? Different forms of mycelium refer various shapes also. Function of mycelium with modification according the circumstance. First one, placentation. Placentation is a fungal tissue. Fungal mycelium hyphae organized loosely or compactly woven to form a tissues are called that is called placentation. It is two types placentation: prosenchyma, pseudoparenchyma. Prosenchyma is these fungal tissues are called loosely interwoven lying more or less parallel to each other. Pseudo is the tissue hyphae compactly interwoven looking like parenchyma tissues cross sections to form. This is the what is the placentation. Coming to the sclerotia. Sclerotia meaning hide. These are hard dormant bodies consist of a compact hyphae protected by external thickened hyphae. Each scleron test germinates into a mycelium on return of favorable condition, that is the penicillium. Another one is the rhizomorphism. See, you can observe the pictures also. Prosenchyma like this. Okay? Prosenchyma, this one. Pseudoparenchyma like this. Prosenchyma, pseudoparenchyma. Coming to the another picture, this is the sclerine. Sclerotia. This picture coming, this one is called the sclerotia. And coming to the rhizoids. Did you understand, girls? Students?
Okay,
Yes?
madam. Yes.
It's not bore. Bore ఏం లేదు కదండి.
బోర్ లేదు మేడం, చెప్పండి.
Okay, good. Next one is a rhizomorphism. Rhizomorphous meaning, see here you can observe the picture. Roots are there, yes or no? White color roots. They are root-like compactly interwoven hyphae with distinct growing tip. They help in absorption and penetration, perennation to tide over the unfavorable period. Example, Armelia melia. This is the rhizome. You can observe the picture. Just root-like structures to present in the rhizomorphism. Coming to the rhizoids. See, this is a mold. These are all the rhizoids. Rhizoid is a short, just root-like structures, very short. And you can observe the root-like structures are there. Here, filamentous is there. The structure you can observe. Structure, filamentous, branch. Yes or no? Not single line. See, here cuts are there. Branch of the thallus generally are formed in tufts at the base of the thallus. They are also function as accruing and absorbing of minerals, nutrient, like this. Okay. The best example, rhizopus. Rhizopus, rhizophidium. Very important. You can observe the where in the in bread mold, in that part, you can observe this is the coming to the uh, Aprisoria. What is another one? Aprisoria. This is the singular is the Aprisorium. Many are there Aprisoria. Okay. Aprisorium. See, you can observe the all are infection. Fungi is not to prepare own food material. No. That's why they are depending other organisms or parasitum, symbiotic like this. That's why this is the infected hyphae. This is the another pathogen depending the dermal. This is the apprecium. This part is called the apprecium. It is a terminal, simple or lobed swollen myceliginous structure of infecting hyphae, which adheres to the surface of the host or other substratum and helps in the penetration of the infection hyphae. See, you can observe the how to penetrate the hyphae infection, other cell organs, bodies. See, this part is where my arrow mark is there. That part is the infection hyphae. These are formed by the some parasitic fungi, such as powdery mildewed in rust. Powdery mildewed rust meaning white rust. Main is a cruciferous family causing this white rust disease. What is the cruciferous? You know, na totakura, the all the amaranthus. In that family, these type of powdery mildews, rust disease observed in this one. Okay. Next one coming to the Hastoria. See, this is the Hastoria. Hastoria meaning or Hastorium. Another name is called Hastorium. One వరకు చెప్పుకుందామా అండి క్లాస్? ఓకేనా?
మేడం ఓకే మేడం.
ఎందుకంటే మరీ ఎక్కువైతే కూడా బోర్ వస్తుంది కదా. 1 వరకు కంప్లీట్ చేస్తా. ఓకేనా? కావాలంటే మిగతాది రేపు సండే ఉంటది కదా. సండే సేమ్ టైం కి మిగతాది కంప్లీట్ చేసుకుందాం. షూర్? ఓకే? ఓకే.
షూర్ మేడం.
యా. Here coming to the next one is a Hastoria or Astorium. Single and singular Astoria. The complex, this one is a Hastoria. Hastorium is an organ. This is a one type of organ. See, you can observe the Hastoria. This is a fungal hyphae. This hyphae where the other plant body, other cells to introduce the hastoryums. Finger-like structures is there, this part. These are the hastoryum to introduce. The organ that develop from a hyphae, usual performing the function of absorption. They are characteristic of obligate parasites. They vary in the shape and may be knob-like or button-like or elongated or fungal finger-like structure or branched. They create some specific enzymes. The hyphae creates from specific enzymes which hydrolyze the protein and carbohydrates of the host plant. This is the Hastoria. These are things I'll explain. These are different from mycelium is the plant body of the fungi. Yes or no? In that mycelium, hyphae, how to form the hyphae, how to form the septae, I'll explain. In that one, how to develop in parasitism, I'll explain. These are steps. Next one, hypal traps. Hypal traps is the sandrus. The another name is called the sandrus. The predaceous fungi. The predaceous fungi develop sticky hyphae or network or hyphal loops known as that is a typical trape and snares are there. They help in the capturing nematodes. See, you can observe the picture. How many nematodes are there in that one? Okay. Stomata. Last one is the stomata. These are compact stomatic structure, much like matrices. The putrefacients are generally formed in them. Okay, this is the last one. The type of mycelium, how to form in that one. That is to explain the important. In general account type, you can observe that these are things also most important. The Thales time. Okay. Coming to the cell structure. Okay. Did you understood total mycelia? Yes?
Yes, madam.
Yeah. Coming to the cell structure. See, first I'll explain general. After that one, I'll explain the in the picture PPT. Okay. Fungal cell wall contain 80 to 90% carbohydrates. The remainder begin proteins and lipids. A typical feature of fungal cell wall is made up of chitin, a prominent constituent of exoskeleton of insects. That is a chitin and not cellulose, the universal cell wall material of plants. Cellulose does not occur in the cell wall. Oomycetes and hypomycetes, the widespread occurrence to chitin. Chitin and cellulose. Chitin and cellulose in the cell wall of the fungi. And relation to the fungal taxonomy has been regulated in the mycologist for about a century. In chitin in the cell wall is main important to made up of chitin. This is the cell wall is the accompanied by the glucose polymerase other than the cellulose mannose. The composition of the cell wall is variable and is modified by the age of fungi and environmental factors also most important for that one. And chitin layer is the most important part of this one. See, characteristic is a main is a what is the important of cell wall. See, this is the fungal cell. Fungal cell. Here you can observe the cell wall is present. Cytoplasm is present. Here in this one ribosomes. This part is the endoplasmic reticulum, mitochondria, lipid inclusions. This one is a cell wall, plasma membrane. In the middle one, you can observe the nucleus. This is very normal cell you can observe to the in the fungal plant body. Here, cell wall determined characteristic. Main is the shape of the structure and shape of the cell. And protection of the osmotic lysis and also act as binding site some enzymes. And what is the chemical composition? Most important chemical composition of cell wall is not the same in all fungi. Chitin is very common, but here five classes is there. Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, Deuteromycotina. These five classes, different orders are there. Each and every order not to same to made up of cell wall chemical structure, but basic one is the chitin is the most important characteristic. Chitin is characteristically present in the cell wall of most fungi. Chitin in fungal cell wall is not strictly identical with animal chitin. Very important this one. The formula of chitin C22H54N4O21 has been suggested for the fungal chitin. The chitin is a linear polymer of N-acetyl beta-glucosamine units linked by beta-1,4 glycoside bonds. The chain may be overlain or intermusted with other glucans like cellulose, which consists of beta-1,3 linked glucose residues. This is the most important characteristic of the chitin. Nuclear in the material in the cytoplasm, you can observe the hyphal tip contain Golgi complex, vesicle called cytosomas, which filled with the cell wall material, and nuclear mitochondria found in contact with the both endoplasmic reticulum, nuclear divisions, intracellular mitosis, karyokinesis, nuclear envelopes remain intact during the nuclear division. Internal spindle developed reserve food material is the main reserve food material. Glycosomes and oils are present. See, here cell wall in each and every class, chitin is the base, no? But here different classification, different types of cell walls are there. Acroisomycetes, acrocymycetes. Here the cell wall made up of cellulose plus glycogen. What is the cellulose glycogen? What type of representative genera? Polysphanolodium, dictyostelium is the main. Another one, Oomycetes group. Taxonomy Oomycetes. Cellulose beta glucan. Cellulose, this cell wall in Oomycetes, the cellulose made up of cellulose and beta glucan is that one. Where the representative genera, Phytophthora indica. This is the albugo. Another common name is Phytophthora albugo, and Pythium is the another one. Pythium is the another part. Next one, hypochytridium tridiaceae. Hypochytridiaceae is the another one. The cell wall made up of cellulose chitin. What is the taxonomic? Rhizidiomycetes. Okay. Another one, Zygomycetes order, taxonomic order. The cell wall to made up of chitin and chitosan. Another thing, the where you can observe mucor and phycomyces, both are there present. Next one is another taxonomic group, Critidiomycetes. Critidiomycetes, Ascomycetes, Deuteromycetes, Basidiomycetes. Observe this one. Here, very dominant class, Cryocycid. Chytridomycetes 1. Second one, Ascomycetes. Third one, Deuteromycetes. Fourth one is a Basidiomycetes. These all classes, the cell wall to made up of chitin B. Chitin B meaning what is a chitin B? Chitin, a linear polymer N-acetyl beta glucosamine with linked with the beta 1,4 glucoside bond. That is the chitin B plus glucan. These all two, the cell wall made up of these two. What are the example? Allo orders. This one is a respective genera. Allomycetes, Neurospora. Here very interesting. Neurospora fungi, very important for the most important for the what are the laboratory use fungi. This Neurospora most important many times to ask this question in the competitive time. What is the main is that one? Polenda. That is a Neurospora. Another one, Aspergillus, forms, Pylophora. Another last class, that is the Ascomycetes class. Mannan, beta, glucan is this Ascomycetes class. Mostly important, Mannan, beta, made up of the cell wall, Mannan, glucans. What is the what is the genera here? Good. Saccharomyces and Candida. Saccharomyces is the yeast. Candida also one type of yeast. Both are the yeast and unicellular is present. This is the total cell structure. Did you understand? Yes.
Yes, madam.
ఇప్పటికి స్టాప్ చేయనా? రేపటి క్లాస్ న్యూట్రిషన్ చెప్పుకుందామా? ఎస్ ఆర్ నో? ఇంకా కొద్దిసేపు చెప్పుకుందామా? Shall I stop?
Okay, madam.
రేపు క్లాస్ లో న్యూట్రిషన్ అది స్టార్ట్ చేసుకుందాం. ఓకేనా?
Okay, madam. Okay, madam.
ఎస్.
Okay,
ఓకే.
thank you, madam.
థ్యాంక్ యూ.
Thank you, madam.
టుమారో సేమ్ 11:15 కి కలుద్దాం. ఓకే.
Okay, madam. Thank you very much, madam. Okay.